some more water for you.
Yeah, of course, guys.
I'll be right back. Wait.
How are you?
Cheeseburger salad? Excellent, there you are.
All right, fantastic. I want to thank everyone for coming. I really want to thank Arthur and everyone else at ACS for the invitation. It's actually really kind of a thrill to be here to chat with you all. This is a fantastic venue, and I hope to be here more in the future. My plan today is to talk to you all about the mechanisms of P2X receptor gating. And it's really going to be a talk that's focused on structural biology and how structural biology determined the molecular mechanisms of gating. And I want this to be somewhat informal. Feel free to ask a question at any point. The talk sort of builds on itself. So if something doesn't make sense at any point, please just interrupt me and I'm happy to answer the question. But before we get into the meat of the talk, I want to give the work a little bit of historical perspective, and then tell a fun anecdotal story as to how I got interested in structural biology. So this always just fascinates me when I think about it, but it's only been about 186 or so years since the concept or the idea that humans and animals were made up of different cell types. It's kind of interesting to think about, only 187 years or 886 years ago. And the idea was first postulated by a German microscopist named Theodor Schwann. And he came to this realization after looking at lots of different organs, tissue organs, under a microscope. And here's some drawings from his seminal work from 1837. And one of the things he noted was that all the different sizes and shapes of what he was looking at under the microscope. Despite that, the one really common theme that they all had was, at least under a light microscope, was, and it's clearly present in his drawings, this darkened border around the outside of these things he was calling cells, right? And he postulated that this was a barrier to ions and to water, and it protected the cells from the environment, the harsh environment. And naturally, the next set of questions arose. Well, if cells have this protective barrier, how is it that they're able to interact with the environment and change their and respond accordingly? And of course, he was correct. This border is, as we all know, the plasma membrane. And this question about how cells can then interact with their environment with such a barrier in place really stumped scientists until 1905, when John Langley, who's a British pharmacologist, postulated the idea that this barrier contains, in quotes, receptive substances. And he said that these receptive substances span from the outside of the cell to the inside of the cell, and that information is transmitted through these receptive substances. And of course, he was correct. We now call these proteins receptors, and there are many types of receptors. There are G-protein coupled receptors, there are ion channels, there are transporters, there are receptor tyrosine kinases. I don't think I really need to dwell on this too much, that these receptors are involved in nearly every physiologic and pathophysiologic process in the human body, from vision to smell to taste to pain, all throughout the cardiovascular system, the immune system, and the central nervous system. They are fundamental and crucial to allow us to live and exist. And as far as the anecdotal story goes, I didn't hear about receptors until college. And I also remember thinking, wow, how do cells interact with the environment? And I heard that these receptors underwent a conformational change. And it may have been from Arthur that I heard this. And I became obsessed with this term conformational change, and I wanted to know, what does this mean? What is a conformational change? So I dug through the literature, the review articles, textbooks, and in every case back then I came across something not unlike what I'm about to show you. The plasma membrane would be drawn as just sort of a rectangle, and the receptor would be drawn as a shape, in this case a circle, okay? And the ligands or molecules in the environment that the receptor interacts with were drawn as smaller shapes, circles with different colors. And in this case, the red circle interacts, the red circular ligand interacts with the circular receptor. And then the magic conformational change happens. And the circle goes to a triangle. And the triangle is able to initiate signal transduction. And that's the extent of what we knew when I was in college. And as fascinating as this was, it was very unsatisfying to me, personally. I had all these additional questions. Why is the circle inactive? Why doesn't that signal? Why is the red ligand recognized by the receptor and not the orange ligand or the blue ligand? Why does the triangle activate the cell? How is it that that's able to initiate signal transduction but the circle is not? And what is the difference between the circle and the triangle anyway? And then, perhaps most importantly, can function of the receptor be manipulated in a strategic manner, right? And that's essentially some of the fundamental questions that structural biology can answer, and one of the reasons why I became interested in structural biology. And the concept is one of the fundamental principles of biochemistry, and that's the structure-function paradigm. And the idea is that the three-dimensional spatial arrangement of all the amino acids, all the atoms that make up the protein, dictate the molecular mechanism of what it is that that protein does, okay? And the mechanism, of course, understanding that mechanism at a molecular level is key to modulating that protein's function. Either promoting its function, if that's what you want to do, or blocking its function, if that's what you want to do. And so, especially as I went through my clinical training, I realized I am a medical doctor. I prescribe medicines. I don't operate on people. And it was also very unsatisfying to me that I prescribed many medicines, and we roughly knew what they did, but not in any kind of molecular detail at all. And so part of what drove me towards structural biology is the idea that if we can understand this for drug targets of interest, we can then strategically design, rationally, agonists that promote or turn on the receptors, or antagonist molecules that specifically block the activation of receptors. And it's incredible to think that if we can understand that and rationally design drugs, we can increase potency of drugs, meaning you take less of the drug to achieve the desired effect, less side effects if you take less of the drug. And also, if you can take a drug that is tailored specifically to the receptor of interest and it doesn't interact with these other receptors, you can get something that's very specific and only interacts with the receptor you're interested in, and that, of course, also limits potential side effects. And so the goal in structural biology is to replace these silly shapes with molecular structures, and high-resolution molecular structures that show us with some detail and some certainty the positions of all the atoms in space. And I do that through two main techniques: X-ray crystallography and single particle cryogenic electron microscopy. And I am going to show you some X-ray and cryo data, and it's going to seem like it was easy to get, but some of the data, especially the crystallography data, took countless hours. And I don't go into much detail as to how these techniques work, but if anyone's interested in that, we can talk about that. And starting about 10 or so years ago, I became fascinated, interested, obsessed with a family of receptors called pure nervic receptors. And these receptors are unique in that they're activated by the binding of extracellular ATP, ADP, and adenosine. And of course, everyone is familiar with how important ATP is to machinery inside of the cell. But Dr. Bernstock in the 70s proposed that ATP actually acts as a neurotransmitter through extracellular signaling. And that was not received well in the beginning, but of course he turned out to be true. It turned out to be correct. And there are three major families of pure nervic receptors, two of which are G protein coupled receptors, and one of which is an ion channel. So the P2Y receptor is a G protein coupled receptor that is activated by extracellular ATP and ADP. And I don't know if some of you have ever heard of the drug Plavix. It's a very highly prescribed drug in the cardiovascular field. If someone gets a stent, they take this antiplatelet agent that helps prevent the stent from closing. So it keeps it open, which is obviously important. The other type of pure nervic receptor is a P1 receptor, or an A1 through A3 receptor. That's the adenosine receptor. So the P1 receptors bind extracellular adenosine. And then we're going to focus today on P2X receptors. So these are not G protein coupled receptors, but these are ion channels. And they respond to extracellular ATP. And it's important to know that there are 7 subtypes, P2X1 through P2X7. And they form trimeric cation channels. And they form homotrimeric channels and heterotrimeric channels. And we'll talk a little bit about that.
Just real quick,
Yeah.
I was curious if ATP, so the P2Y is actually, it's basically it's fixed open by plavix to allow ATP to constantly flow in.
So we'll take a step back. So P2Y and P1 receptors are G protein chemical receptors. They're not ion channels. So no ions flow through them. The way these work are ligands bind to them, and then there's a magic conformational change, which is now well understood. But the receptor, which spans the plasma membrane, changes its shape and opens up a pocket that allows what's called a G protein to bind there, and that activates a signal transduction pathway. So ATP initiates that conformational change to allow the G protein to turn on machinery inside the cell. P2X receptors are ion channels, so they recognize extracellular ATP, and actually you'll see this mechanism later on. ATP will bind, and a pore will open that allows sodium to enter the cell, calcium to enter the cell, and potassium to efflux out of the cell. But it generates a net inward current, and I'll show you that in a couple of slides.
Yep. Just a quick piece. Is the adenosine AMP, or is that another? Is AMP or cyclic AMP another?
So it's not. This is not cyclic AMP. This is straight adenosine.
Just straight
Yeah.
adenosine. Okay, just there.
Nucleotide. Yep. Okay, so now why does a cardiologist, right? Arthur mentioned that do clinical. I have some clinical training, and I do see patients in cardiology clinics. So why is a cardiologist interested in P2X receptors? Well, it just so happens that each of the different subtypes have important roles in a number of cardiovascularly and central nervous system related processes, such as platelet aggregation, vascular reactivity, ischemic pain, which is the pain that happens when a person has chest pain from lack of blood flow to the endocardium of their hearts, vascular inflammation, which leads to coronary artery disease from plaque buildup, chronic inflammation, and atherosclerosis. So these subtypes are very, very clinically relevant and important to a cardiologist or anyone interested in the cardiovascular system or the central nervous system. And just some of the biochemistry of P2X receptors, I mentioned that they are trimeric proteins. So what that means is 3 polypeptides come together to form one functional receptor. And they form both homotrimeric and heterotrimeric receptors. So what a homotrimeric receptor means, so for example, P2X2 receptor, it means 3 protomers of P2X2 come together to form one P2X2 homotrimeric channel. Or a homotrimeric P2X3 receptor means the amino acid sequence that forms the polypeptide chain of P2X3 receptor, 3 of those come together to form a channel. Heterotrimeric channels means that some subunits of P2X2 and some of P2X3 come together to form a mixed channel. And these are actually, very little is known about these, and they have unique properties that are just different and distinct than the homotrimeric channels. And it's actually not known whether it's, because these are trimeric, whether it's 2 copies of P2X2 and 1 copy of P2X3, or 2 copies of P2X3 and 1 copy of P2X2, or a mix of both. And this is a ligand-gated ion channel, where the ligand is ATP. So when ATP binds, the channel opens, sodium and calcium enter the cell, potassium effluxes out of the cell, and it generates when the receptor gets activated, it mediates a net inward current. And I will show you next. The P2X receptors have very, very interesting pharmacology, very interesting biochemistry, and some very interesting properties, which I'm about to go over with you. So as far as the pharmacology goes, the different subtypes respond across a wide range of extracellular ATP concentration. So their affinity for ATP is quite different. So P2X1 and P2X3 receptors, it only takes roughly 1 to 10 nanomolar concentrations of extracellular ATP to turn them on. P2X2 and P2X4 receptors take a little bit more, 50 to 100 nanomolar roughly. And look at P2X7 receptors. It takes orders of magnitude more extracellular ATP to activate the channel. And nobody really knows why this is. We have some ideas, but we haven't proven it, but nobody knows why that is. So it's very, very interesting. Very unique and wide-ranging pharmacology. But they also have what I'm calling differing gating kinetics. And I'll tell you what that means in a minute. But there's a principle of ion channels, ligand-gated ion channels, called desensitization. And I'll show you an example and show you electrophysiologically what that means, but just to explain it, it means that when ATP binds the receptor, some of the receptors turn on, they open up, but they only open for brief moments of time. Even though the signal to stay open is present, there's extracellular ATP, they are not open for very long. In fact, P2X1 and P2X3 receptors desensitize after milliseconds. So they're only open for a few milliseconds, tens of milliseconds. P2X2 and P2X4 desensitize over tens of seconds, 30 seconds, 60 seconds. And P2X7 receptor does not desensitize at all. So as long as there's the appropriate concentration of extracellular ATP present, the channel stays open. Okay? And we're going to focus on why this is today. I'm going to show you what I mean electrophysiologically. So here is a cell that is expressing P2X1 receptor. Okay? And when it's exposed to extracellular ATP, evidenced by this black rectangle, you can see that there's this downward deflection that represents a net inward current. And if you look at the time scale here, this is the time scale bar for 20 seconds. So it's only open for less than a second. And it immediately closes, even though the signal to stay open, ATP, is present until all the way over here. So that is called fast desensitization. And if you look at the other subtypes, P2X3, as I alluded to, looks a lot like P2X1. It's only open for a few milliseconds. But look at P2X2 and P2X4. So they open and they slowly close over time. So as long as they're constantly exposed to ATP, they'll eventually nearly completely close in the case of P2X2 and pretty much completely close in the case of P2X4. And if you look at P2X7, as long as ATP is present, the channel stays open. It will not desensitize. And so the question that I asked 10 years ago or so now is, what is the structural mechanisms underlying desensitization? Why is it that some P2X receptors desensitize in milliseconds, some desensitize after 60 seconds, and P2X7 doesn't desensitize at all? And people had hypothesized that P2X receptors desensitize through a ball and chain mechanism, which is similar to potassium channels, where literally a clump of amino acids effectively plug the pore. We didn't think so, and so we wanted to prove that. And so I chose to focus on P2X3 receptor because it desensitizes rapidly and easily. My thought was, if I could understand the structure of the desensitized state, I might be able to explain desensitization. And then the project sort of expanded, and I thought, well, what are all the relatively stable conformations of this receptor? And if you think about it, there are at least three, and probably just three, and we're going to step through them here. There's a closed apo resting state, meaning the receptor is not exposed to any ATP. So you would imagine that there would be no ATP present anywhere on the receptor, and the pore, because it's an ion channel, will be closed. So sodium cannot enter. Okay? So that's, and we'll represent that with a circle. Another conformation would be when you expose the circle to ATP, the channel should open and there should be ATP bound to the receptor. And we'll call that a triangle. So the ATP bound, open, active state. And then finally, the square should be what we're calling a desensitized state. But if you think about what that means, ATP is there. So ATP should be bound, but the pore should be closed. So three conformational states. In my imagination, my wish, my hope back then was to solve the structure of all three of these states. And if I can do that, by looking at the molecular structure of the receptor in all those states, I could deduce the mechanism of desensitization. That was my plan. And I wanted to do that using X-ray crystallography. So hours and hours, I can't tell you how many hours it took to get crystals. And when you're a crystallographer and you walk in to the 4-degree room, which is where I set up, I spent hours in a 4-degree room setting up crystallization trays and harvesting crystals, and you see things that look like this, this is eye candy. These are protein crystals of P2X3 receptor, and they're incredibly beautiful. But one thing you should note as a crystallographer, the beauty of the crystal does not tell you how well it diffracts and will not tell you how good your data is, unfortunately. But still, you get excited when you see things like this. And this crystal happened to be one of the magical ones. But that crystal produced this diffraction pattern, and we don't have time to go into what this means but this diffraction pattern is unique to the positions of all the atoms in space that make up that crystalline lattice. And this little red circle represents the 3 angstrom arc. So the concept is the farther out to the edge you see spots, the higher the resolution, the more certain you are about the positions of atoms in space. And there are spots that go beyond 3 angstroms. So this was a 2.8 angstrom data set, which is absolutely. And so right away, I was able to change this circle into a structure of P2X3 receptor in an apostate. And almost immediately after that, I was able to replace this square with the desensitized state structure. And we're going to go into this in more detail, but the keen observer might already start to see some differences in the transmembrane domain, which we're going to talk about in a few moments. But how can I get the structure of this triangle, right? I mentioned to you that as soon as you add ATP within milliseconds, the channel desensitizes. So how do I capture an open state? And I struggled with that for a while until this, unfortunately it's cut off, but this beautiful paper by Hausman showed that one can convert a fast desensitizing subtype into a slow desensitizing subtype, and conversely, a slow desensitizing subtype into a fast desensitizing subtype by making a few key mutations in the N-terminus. And these aren't random mutations. So it's, in the case of P2X3 receptor, it's mutating a threonine at position 13 to a proline, a serine at position 15 to a valine, and a valine at position 16 to an isoleucine. So where on earth did they come up with this from? I'll tell you in a moment, but here I've shown this before, and this is how fast P2X3 desensitizes, right? You expose it to ATP, it's on very, very briefly. You make these mutations, and it slows down desensitization, and in fact there's a little bit at the end that never completely desensitizes. And what they did here is they made the mutations that corresponded to the analogous subtype. So it's a chimeric receptor that's 97% P2X3 and has 3 amino acids that are found in P2X2. So you can convert P2X2 to slow by making these mutations found in P2X2, and you can turn P2X2 from a slow desensitizer to a fast desensitizer by making the corresponding mutations in P2X3. So it's telling you right away that the mechanism has something to do with these 3 residues. Okay? And importantly, when I made this chimera protein, it didn't affect the affinity for ATP in this case. And so this is a radioactive ATP binding experiment. On the x-axis is increasing amounts of ATP, and on the radioactive ATP, and on the y-axis is a radioactive signal. And you can see that the radioactive signal increases until there's no more ligand binding sites for the radioactive ATP to occupy, and then it saturates. You can get a KD from that, so it tells you that the affinity is about 3 nanomolar, which is about what the published affinity is for P2X3. And when I made the chimeric receptor, it's again about 3 nanomolar. So that this mutation does not affect the ability to bind ATP.
Sorry, I was just asking, did you say that that modification would be made to any of those types of proteins, not just this type of P2X?
So what the paper showed was you could convert P2X3 from a fast desensitizer to a slow one, and P2X2 from a slow desensitizer to a fast one by swapping these three amino acids, and they aren't random. Remember, they're the ones that exist in the opposite partner. So 2 to 3 and 3 to 2.
Do you know if that's been observed in other types of?
So no, great question. I mean, no one's done it. It's a great question, but I just pursued this chimeric receptor because I wanted to try to capture an open state structure. Oh, fantastic. So I got lucky, and that chimeric receptor produced an ATP-bound open state structure. And from now on, throughout the rest of the talk, anytime you see anything in this reddish color, that's the apostate structure for stop. The green structure is the ATP-bound open state structure for GO, and then the yellow is the ATP-bound desensitized state structure. And we're going to be seeing lots of models coming up that show you exactly how the channel opens and desensitizes. And you can already probably start to see some differences in the transmembrane. So I want to orient you all to the overall architecture of a P2X receptor. Okay, remember it sits in the lipid bilayer. It has an extracellular domain, a large extracellular domain. So this is outside of the cell. It has the extracellular domain. It has a transmembrane domain. And if you remember what I told you earlier, 3 protomers come together to form a functional channel. Each protomer contributes 2 transmembrane domains. So the overall receptor has 6 transmembrane domains. But we refer to the more N-terminal transmembrane domain is transmembrane domain 1, and the internal pore lining, more C-terminal transmembrane domain is transmembrane domain 2. Okay? So the N-terminus is somewhere here. It passes the lipid bilayer, extracellular, a lot of residues, and then it comes back across to form transmembrane domain 2, and then there's a cytoplasmic domain down here. The one thing you'll notice is that in this model, we don't see any residues in the cytoplasmic domain, and that becomes a theme that we'll talk about a little bit later. But, so, there are 3 transmembrane domain 1s, 1 from each protomer. There are 3 transmembrane domain 2s, 1 from each protomer. And just take note of how transmembrane domain 2 lines the inner portion of the domain. So that's the pore lining transmembrane domain. So the pore is where ions flow. So when ions flow, they're going to be interacting with transmembrane domain 2, not transmembrane domain 1. Okay?
Do they just shoot right through it? Or do they,
I'm sorry?
Do they just shoot right through it? Or do
You'll
they
see.
like violent?
Yeah, you'll see. Yeah, I think you'll see. So I told you that this is an apostate structure. Why should you believe me? Right? So we'll step through. What is an apostate structure? There's no ATP in the extracellular domain, and there's a magnesium, but in this density there was no ligand density for ATP, so there is no ATP. And the pore ought to be closed, right? No ions should be able to flow through it. So if you imagine putting your eye right there, right on the extracellular part, so that you're going to be seeing what an ion would see. We're going to rotate this 90 degrees, and we're going to look straight down the pore from the extracellular surface. This is a space-filling model. There's no room there, right? There's no room for anything to enter through here. This pore is closed. So there's no ATP and the pore is closed. This is an apostate structure, okay? Now, I told you that this is an open state structure. Why should you believe me? Well, there's ATP present in the extracellular domain, and if we look down the pore, there's a hole down the middle of the pore, and the hole is large enough to pass partially hydrated sodium ions, which is exactly what P2X receptor does. Now, interestingly, this is a desensitized state structure. So you can see right away there's ATP in the extracellular domain, which is what we would expect. And for desensitized state structure, we expect the pore to be closed. And if we look down the barrel, it is closed. That is too small, and an ion cannot pass through that small little surface there. And so here's the scheme, and the data was so good, we could actually pinpoint exactly which residues formed the constriction site to block sodium from entering, and which residues interacted with sodium as it passed down the pore. So we're going to look at each one of these in some detail. The apostate structure, the ATP-bound open state structure, and the ATP-bound desensitized state structure. So, and if we look carefully at the structure, we can start to deduce some interesting features, okay? So, to orient you, you're looking down the pore. This is the path of the ion flow. These outer transmembrane domains are transmembrane domain 1. The pore lining transmembrane domain is transmembrane domain 2. So you can see that there's 3 of them, and you can see specific amino acids in our model. The data was good enough to say that isoleucine 323 is the constriction site. That's what's blocking. They're so close in this pore that they're physically blocking sodium ions from flowing. And one of the things I want to point out is I think we all learned in biochemistry that a perfect alpha helix is 3.6 residues per turn. And I'll just tell you that this is a perfect alpha helix. You can just look at it. It looks like a perfect spring. If you take a look at the ATP-bound open state structure, though, you can see what happens here. Look at transmembrane domain 2 now. It is no longer a perfect alpha helix. It has been stretched in the center. It's what's called a 310. It has a stretch of a 310 helix. And so I liken this to a spring that is fixed at one end, fixed at this end, and pulled at the other end. It looks like a stretched spring, which is exactly what that looks like. And then, if you look at the desensitized state structure, it goes back to a perfect alpha helix. So it is stretched and then recoils back, transmembrane domain 2. And I want to show you all, this is a movie. It's an interpolation between all three of the states. So again, you're looking down the path that an ion would flow, and you'll see transmembrane, this is the apostate structure, you'll see how this gets pulled and stretched. Now there's a threonine residue with the oxygen pointing in the middle to interact with the ions, and then it desensitizes by recoiling back, and then there's a valine residue that forms the new constriction site. So apo is closed and desensitized is closed, but they're closed in different ways. Okay, we'll look at that one more time. We're looking down the path of the ion flow. Isoleucine 323 prevents ions from flowing. Oops. I think it's not going to want me to do this from here unless I go back. There we go. The helix gets stretched from the extracellular end. It pulls transmembrane domain 2. The threonine is able to interact with ions, and then it recoils to the valine that forms the constriction site, and then it resets. And nobody understands quite how it resets. But that is the motion that happens when the pore opens. Yep.
How thick is this relative to the membrane? Is this like a camera aperture that we're
Yes,
talking
it's
about?
a lot
here?
like an aperture.
So it's one little gate in the middle of
Yep.
the membrane
Yep.
thickness.
Yep. And that's a great analogy. And a transmembrane domain spans the entire helix. So you'll see that, and make note of this, and I'll point this out later, that in the apo state, the constriction site is more extracellular. And in the desensitized state, the constriction site is deeper in the membrane. And I think we're about to get into some of that. But here, again, in reddish maroon is apo, and in green is open. And you can see that what happens if you align these two structures, you can see that what happens is there's a movement of the extracellular portion of transmembrane domain 2, the extracellular part. And when the receptor goes from the open state in green to the desensitized state in yellow, there's a movement at the cytoplasmic surface. So to open, there's a movement at the extracellular surface. To desensitize, there's a movement at the cytoplasmic surface. And so the question is, why does TM2 rotate to desensitize the channel at all? Okay, why doesn't the whole helix just move? And to point out what you're saying is, if you look carefully here in the apo state, you can see that the transmembrane domains look a lot like a teepee in this state. And it looks more like a cone in this state. And one of the things you also might quickly notice is, even though the residues are present, we can't see any of the cytoplasmic domain in the apo state, and we see very, very little in the desensitized state, but we see a lot more in the open state. And in structural biology, the common rule is, if you have residues present but you can't see them in the structure, it either means it's unstructured or it's not homogeneous. There are multiple conformations. And so it ends up getting averaged out and you can't see it. And so what I want to focus on is these residues that we can now visualize in the open state, which we couldn't see in the apo state or the desensitized state. So I highlighted this box. We're going to take it. We're going to magnify in on this box. Yep.
So the forces that move these things around are generated in the extracellular.
Yep. I don't have time to talk about it, but I'll show you. So when ATP binds here, P2X receptors are named according to the scheme, a nautical theme, because one of these protomers, and you'll see it later, looks like a dolphin sort of jumping out of the ocean. And so things are named according to anatomy of a dolphin. So there's a head, and a flipper, a left flipper, and a right flipper. And in any case, ATP binds between the head domain and one of the flipper domains, and they kind of close in on each other. And when that happens, there's these beta strands, you can't really see them, that attach directly to transmembrane domain 2, which is the pore lining transmembrane domain. So when this lever moves, it pulls on the extracellular surface here to open and stretch open the transmembrane domain? Good, great question. I want to look in on the open state structure because we can see some residues here. We can see in the model they're now visible, and they weren't visible at all in the aqueous state. And you can see there's just a little bit of a hint of this additional helix in the desensitized state that you can't really see in the aqueous state. So I wanted to focus in on this because this might be telling us something about how and why this channel is open. And so the magnifying is colored a different color green. So there's forest green, regular green, and light green. And the 3 mutations that I made, so T13P, so the mutation to a proline is shown here in yellow, S15V with valine is shown here in yellow, and V69 is shown right here. They're all interacting together, and if you flip this hydrophobic patch forms here. We called it the cytoplasmic cap, and the pore is stretched open from the extracellular domain. And as long as this is present, the helix can't move. And the thought process is, if this breaks, and this is your spring that is stretched from this end and fixed from the cytoplasmic end. And once this breaks, is if you let go, the helix recoils back and forms more of a cone. So it goes from a TP to a stretched helix, recoiling to a cone. And so I call this the helical recoil model of receptor desensitization. Very unique mechanism, never before recognized in any other ion channel. So it was a very kind of exciting model for us. And so how does this work? So the idea is P2X1 and P2X3 I mentioned are fast desensitizers. So the thought is, my hypothesis is correct, they have an unstable cap. So the cap might form, but it doesn't stay there for very long, so it breaks, and the helix recoils very quickly or almost immediately in milliseconds, and the receptor desensitizes. So P2X2 and P2X4 desensitizes slowly. It has a more stable cap. It stays around longer, and the longer that cap is there, the longer the channel is open. So if I'm correct then, what about P2X7 receptor? It must have a permanent cap, right? It must never break, if my hypothesis is correct. So that was the next question that I wanted to pursue, is I really wanted a structure of P2X7 receptor. And my thought was, if I could solve the structure in the apo state, and I saw the cytoplasmic cap present, I believe that the helical recoil model of desensitization would show support for that model. So I desperately sought a structure of P2X7 receptor. And I tried, so is the cytoplasmic cap formed in the apo state of P2X7 receptor? That's the main question. And I tried and tried and tried to get crystal structure of P2X7 receptor, and I failed and failed and failed. And this was right around the time where single particle cryo-electron microscopy was sort of having its resolution revolution, where it really started to improve data quality, and you started seeing data that was rivaling crystallography. And I thought, could I potentially use cryo-EM to solve the structure of P2X7 receptor? And so after extensive failures, I realized that P2X7 receptor is the largest of all the P2X receptors, and one of the themes is in cryo-EM that the larger your molecule is, the better it is that you might be able to get quality data. So that was in my favor for trying to switch over to cryo-EM. And also in the literature, it suggested that there were putative glycosylation sites, a lot of them in P2X7 receptor, and these putative palmitoylation sites that would contribute to heterogeneity that probably prevented me from ever getting a crystal to begin with. And so I sort of moved on and tried to do cryo-electron microscopy. Okay? And when I did that, I saw the following. What you're looking at here is you're looking at purified P2X7 receptor reconstituted into a detergent micelle, suspended in a thin layer of vitreous ice, and magnified. The image you're looking at is 150,000-fold magnification of protein suspended in a thin layer of ice. And you might be able to immediately recognize something. When I saw this, I was very excited. So what you're looking at here is individual protein particles, and here is a receptor. And you're not going to see anything more clear than this, believe it or not. So let's just step through this. In the center, you see this. You see kind of 3 different domains, right? In the center is this oval thing. That is the detergent micelle. The transmembrane domains are inside that thing. And this larger piece here is the extracellular domain. It's big, and here the cytoplasmic domain is smaller. But look, you can see something on both sides of the detergent micelle. So when I saw this, I immediately knew I am going to see density, meaning the model. When I build the model, I'm going to see things I've never seen before. I'm going to see this entire domain. So I very quickly collected data, and what you do with cryo-EM is you take thousands and thousands of these images, and what you're looking at here are particles. These are 2D projections of particles that are part of the little proteins, suspended in a thin layer of ice in multiple orientations. So exactly like if you think about your shadow cast from the sun on the ground, depending on how you stand, it casts a different 2D shadow. Well, that's what these are. These are 2D shadows. And sometimes there are side views like this, and sometimes there are more top-down views like this. And what you do is you collate and collect thousands and thousands of these individual particle images, these projections, and a computer can help you collate them into what are called 2D classes. These are all the various orientations of these 2D projections. And so you can see these incredible side views where, so you can see the micelle. I don't know if you all can see that X that's in the middle there in bright white. That's the transmembrane domain. Here's the extracellular domain, and here's this beautiful cytoplasmic domain that I'd never seen before. And when I saw these 2D classes, I knew I was on something. And then in cryo-EM, what you do is you take these 2D classes, you give it a starting model, and then the computer can help you put together these 2D projections to back project them to form a 3D object. And so you get what are called 3D classes. And sometimes a lot of the particles are what we call junk particles, meaning they aren't the best quality. And the computer can help you sift through them until you get a 3D model. And you can just see, even if you don't know much about structural biology, you can see that this just has more features. Like in these helices, you can see that there are grooves and ridges in these helices where these look more like tubes. So the lower the resolution, the more tube-like. The higher the resolution, the more groove-like. So I selected this 3D class and then moved on to do what's called iterative. It's a refinement, which is an iterative process where the computer helps you align all the particles and all the different 2D and 3D projections. And the more you align, the higher and higher resolution you get and the more features you see. And so I ended up with a 2.8 angstrom resolution map of P2X7 receptor, and here it is. Each protemer is colored in a different color. And so it has the same themes as what I've shown you before, the large extracellular domain. This is an apostate structure, so there's no ATP, but ATP would be bound right here if it were an open state structure. Here's the transmembrane domains in here, and here's this amazing cytoplasmic domain, which I had never seen before. And we're not going to talk much about it, but it was chock full of some very incredible findings that we're pursuing now. But one of the things you might notice is these maroon, long tail-like things, and we'll talk about those in a minute. And so here's what the model, here's what this model looks like. And you can see, there's the cytoplasmic cap. It's that nested beta strand structure, and it's present there in the apostate of this receptor. And so, if you take, and this is sort of the dolphin. This is kind of a dolphin jumping out of the ocean. So what you have here is one protemer of P2X3 receptor shown here, colored by domain, and one protemer of P2X7 receptor on the right, colored by domain. And so you can see that the extracellular domain is in blue. There's an ATP molecule in the open state structure of P2X3. There's no ATP. Apostate of P2X7. Each has transmembrane domain shown in green. The cytoplasmic cap is shown. This is the part of the molecule where I made those mutations. That's shown in cyan, and you can see it's present in both. P2X7 has this large, unusual cytoplasmic domain. Let's set that aside for a moment. But look at this. P2X7 has this additional 18 to 20 residues in purple here. So there's a cytoplasmic cap. That I called the C-cis anchor. It's present in P2X7 receptor, but it is not present in any of the other subtypes. And if you look at where it is, it connects transmembrane domain 2 to the cytoplasmic cap. It physically has 18 residues that sit in between those two important domains that I've mentioned are responsible for gating. So the question is, how does this affect the gating of P2X7 receptor? And when you look at it, so here's transmembrane domain 2, here is the C-cis anchor, 18 or 20 residues, and here is beta 15, which is part of the cytoplasmic cap. One of the things you'll notice right away is that there are a lot of cysteine residues in this C-cis anchor. And in the density the model I was building, I saw a lot of long linear densities that kept appearing off the edge of the cysteine residues. And what that is palmitoylation. So it's a post-translational modification to the receptor on one serine residue, which is unusual, and a number of cysteine residues. And I immediately thought, oh, that's why this receptor doesn't desensitize. So that was my hypothesis. If it's palmitoylated, and if you look carefully, the palmitoyl groups are pointing back toward where the plasma membrane would be. They are locking and linking transmembrane domain 2, which is the transmembrane domain that moves. They're locking it in place, preventing it from undergoing the recoil that desensitizes the receptor. That was my hypothesis. So I thought, well, what if I just physically remove these 18 residues from P2X7 receptor? So TM2 appears to be anchored in place. That's why I call it the CSIS anchor. And here, in black, is an electrophysiological tracing of P2X, wild-type P2X7 receptor. It doesn't desensitize. If I just simply remove the CSIS anchor, shown in red here, you can see that the channel starts to slowly desensitize. I was never satisfied with removing 18 residues from the middle of this, this complex receptor, so then I went through and I mutated out all the cysteines. And when I did, it kept them, mutated them to alanines. So it keeps the register of the receptor, but alanines can't be palmitoylated. And when I do that, it very briskly and very nicely desensitizes.
Steve, did you just mutate those in the cap, or did you do everything?
No, I mutated these in the anchor.
Okay.
So these 7 or 8, and then that's serine. So one serine and 7 or 8 cysteines. Mutate them all to alanine, and it desensitized. So the overall big picture idea here with the helical recoil model of desensitization is you have so P2X1, P2X2, 3, 4, 5, and 6, but yeah, 1 through 6. They in the apostate, they're shaped like a TP. ATP binds. The cap is not formed. It dynamically forms because of this conformational change. It fixes the receptor in the open state until it breaks. And once it breaks, the helix can recoil and form a desensitized state structure. That's the helical model of receptor desensitization. But P2X7 receptor has these palmitoylated groups that all it can do is undergo the extracellular conformational change. It cannot recoil because these palmitoyl groups are locking that transmembrane domain in place, making it uniquely among all the P2X receptors not undergo desensitization. The last little bit here I just wanted to kind of tease you is part of why I'm doing this is I am interested in molecular mechanisms of how these ion channels work, but part of my drive as a clinician is to develop small molecules that back to the beginning, like why does the red ligand bind to the receptor? Why does that red circle bind to the receptor? I'm interested in understanding the molecular pharmacology is the term I use. And so I want to do it as a means for drug development. And so here's what I mean by that. So in the blue mesh here is the data we see that represents the ATP molecule and the pose that it sits when it's bound in the pocket. And you can see here that we've nicely fit ATP into this density. And this is the only way that ATP can fit inside this density and still satisfy the criteria for carbon-oxygen, carbon-carbon distances, and the restraints that are conferred on the ATP molecule. And what's nice about it is you can see exactly how each of the residues in P2X3 receptor interact with ATP to allow it to bind there and activate the channel. And so, yeah, we refer to this as molecular pharmacology. And the goal is to try to understand for agonists and antagonists what is the molecular pharmacology of those various binding pockets, so we can then design drugs that either turn it on or block it from being turned on. Here, I just want to show you that here is the ligand pose. That's what we call the way ATP binds in the pocket. Here is the ligand pose of ATP bound to P2X3 receptor, solved by me in 2016 using X-ray crystallography. So you can see exactly the pose, you can see all the residues and how it interacts with that molecule. So here's the molecular pharmacology of this binding pocket. Here is the same thing in the P2X7 receptor, solved by cryo-EM in 2019. solved by me, and I can say that I did not cheat. I solved them independently using the density and the restraints at hand, and look at how stunningly similar the pose of ATP is in the binding pocket, and all the interesting residues. A lysine interacts with the gamma phosphate, a lysine interacts with the gamma phosphate, an arginine interacts with the gamma phosphate, an arginine interacts with the gamma phosphate. They're almost identical. It almost makes you believe science is real. And so we are trying to do the same thing for other agonists and antagonists in order to design more tailored drugs. So just to summarize, I really want to convey this idea of the structure-function paradigm and how fundamentally important it is to biochemistry and to structural biology. And then just to mention P2X receptors and how important they are to a lot of physiologic and pathophysiologic processes in the central nervous system and the cardiovascular system. So specifically, I'm very interested in vascular inflammation. Vascular inflammation is one of the causes of atherosclerosis, so as a cardiologist, that really interests me. There's a lot of, I'm also interested in the central nervous system and neuroinflammation and overactive P2X7 receptors and implicated in neuroinflammation and disease processes of neurodegeneration, as well as things like systemic hypertension and platelet aggregation. So very, very central to what I do clinically. And I just want to remind you of the P2X receptor mechanism of desensitization, the so-called helical recoil model, and how P2X7 uniquely does not desensitize because it has these extra 18 residues, many of which are palmitoylated, to prevent that helical recoil. So it keeps the channel open and prevents it from desensitizing. And then just, you're going to see in the future drugs that target P2X receptors, and we're hopefully going to be developing some of these compounds through what we call rational drug design. I have a lot of people to acknowledge. I have a fantastic group. I sort of semi-independently started a lab in 2017. Alana was my first research assistant, and she's now in medical school. Now I am fully independent with my own lab space, and I have a wonderful group, a mixture of graduate students, a master's student from Germany, and some fantastic research assistants that are from Reed College, and it's a fantastic experience working with them, and we're making some good progress. Of course, I want to acknowledge Arthur. I can honestly say I really wouldn't be here if not for Arthur. So he really impressed, when I was an 18-year-old kid, he impressed upon me and he reinforced my love of science. And so I can honestly say I wouldn't be here if not for Arthur. And Eric Lowe was my postdoc mentor. Dave Ferens was my PhD mentor. And Rouge Walsh, she is one of the collaborators that I have at OHSU. And I have a number now of international collaborators from around the world that are providing some of the drug development studies. And I'd like to thank various funding sources, including the NIH and Gilead Pharmaceuticals. And I want to thank you all for your attention, and I'm happy to answer any questions that you might have.
Before the questions, is anybody still hungry? Did anybody not get their food?
Yeah. So in the P2X7, where you have the palmitoyl groups that stabilize it in the open position, once you remove the ATP, it does close.
Correct, because it closes from movements on the extracellular surface, right? So if you go back to this, it just goes from this back to this. So no movement ever occurs down here. It's all, when the ATP leaves, it undoes the conformational change, the lever that I was talking about that pulled on the extracellular surface of transmembrane domain 2. So it just flip-flops back and forth between these two states. And one thing I will say, the P2X7 receptor, at least electrophysiologically, is if you get a nice stable recording, you can just put on ATP and put off ATP, and you'll just get a nice steady current. It'll just turn on to the same current level as before, where some of the other receptors that doesn't happen because it takes a long time to undo the desensitized state, and nobody really understands how that gets reset, and and so it's kind of difficult to work with the receptors that desensitize here.
So if the binding pockets for ATP are so similar between P2X 3 and 7, what's the reason? Is it known why the binding affinity is different?
So let's see, I think I included this somewhere. So here's why I think it is. I don't have proof of this, okay? But this is what I think. So here is the.
Oh, you're fine.
Here's the apostate structure of P2X3 receptor. Each protemer is colored a different color. So light, dark gray, and red. Oh yeah, watch the cord. Yeah. Here is the path, the entry path of ATP into its binding pocket. It's wide open in P2X3. Here's the apostate structure of P2X7 receptor. Look at the path of entry into the binding pocket. It is much, much more narrow and constricted. So I think, I have to find a way to prove this, but I hypothesized this in the last paper, that it has to do with access to the pocket. It is much more limited, because all the interactions are the same. Once it gets there, it makes the same interactions. But it can't physically get there as easily in P2X7 receptor as it can in P2X3 receptor. So that's what I think it is. So it's access time. And so you effectively need a lot more ATP here to get in there. Good question.
So do you see heterotrimers forming between like all of the different units? Okay.
No, so yeah, another good question. That's what we're so it tends to be, so the most prevalent ones are 2 and 3, and then 1 and 4 I would say is the second most popular.
So 2 and 3 are like the faster and slower desensitizing, so do you see intermediate?
Yeah, so heterotrimers have a completely unique desensitization and gating profile, distinct from either. Yeah, nobody understands why. There are no structure. It's very hard to do. And the reason why it's very hard to do, I'm sure get into this, sure. So in cryo-EM and in crystallography, there's this concept of symmetry, okay? And when you have, go back, when you have something like this, when you have something like this, right? The computer is helping you align these particles, or it's doing it for you. And you give it some guidance, but it's doing it for you. But when you have symmetry, it helps it to align, okay? And it actually improves your signal noise, because if you have one particle, you have 3 particles because it's threefold symmetric. What if you have something that's so-called pseudo-symmetric, where they're very, very close to threefold symmetry, but they're not quite? The computer can't differentiate those, so it ends up averaging them, and you get something that you can't interpret. So for heterotrimers, you have to break the pseudo-symmetry. And the way to break the pseudo-symmetry is to find a molecule that will bind to P2X, that's large enough, that will bind to P2X2, but not 3, or 3, but not 2. So you end up getting, instead of something that looks like this, you'll see something that has a couple of nubbins sticking off of it. So the computer can help align. We're desperately trying to do that through various techniques, but it's not easy to find either antibodies or these things called aptamers and nucleic acid sequences. We're trying to do that because we're interested in P2X2, 3 heterotrimers and 1, 4. We may have a way to do 1, 4, maybe. Yeah.
So when you're talking about a cytoplasmic cap,
Yeah.
you said that there's a sort of, sorry, because you can run the structure of that, sorry.
Sure.
There was a region that was like, holding the P2X7 as opposed to P2X4 and 2. But there's also sort of a lot of extra stuff happening.
Where? Down below?
Yeah.
In P2X7?
Yeah.
Yeah. Yeah, so
Where
it has this
the
unique
functionality?
Oh, great question, yeah. Yeah. So all this stuff.
Yeah, all that.
So P2X7 receptor has an extra, I don't know, 200 and something residues per protemer than any of the other ones, and that's why we were able to see it. And found within it are 2 zinc ions and a guanine nucleotide that purified out of mammalian cells as a GDP molecule, and we proved it through various mass spec methods. And we're trying to figure out what it does. And so what is interesting about this is, for years, if you read the literature, I had mentioned G-protein coupled receptors earlier. The buzzword that people say is that G-protein coupled receptors have metabotropic function, and ion channels have inotropic function. Yeah. People for years have said in review articles and literature that P2X7 seems to have both inotropic function and metabotropic function. And so it's my hypothesis, and we don't know if this is true, but that it confers some of its metabotropic function through this domain, through interacting with a number of other proteins. And you can find people talking about 30, 40 proteins that interact with the
I was gonna say, so like in what proximity, like if that's the hypothesis, in what proximity are you looking at this in terms of other membrane proteins?
Oh, yeah, I don't know about other, I don't know, I don't know that it's other membrane proteins. I think it could be other cytosolic proteins, soluble proteins inside the cytoplasm. And one thing I will tell you is that something that P2X7 receptor does when it becomes activated is it can initiate apoptosis. If you remove the cytoplasmic domain, it will no longer initiate apoptosis. It will still function as an ion channel, but it will not initiate apoptosis. Yep.
So you mentioned the 7 has this cap and the thing in the domain, but I thought you said the other ones have a cap that you couldn't see by X-ray crystallography?
Yeah. So P2X3, so the idea is they have a cap. They all will have a cap in the open state. Set P2X7 aside. So essentially what I'm saying is we couldn't see it. We couldn't see it.
Yeah, is that possible that you could see it using like the cryo-EM on those
We've done that.
Instead?
Well, I'm not going to talk about that yet, but yeah, we don't see it. We don't see it in P2X3. Yeah. Only in the open state.
So, sorry, just to keep following up on this line. Yeah. So are you, without that cytosolic extra stuff, are any of these
these receptors, P2X3 doesn't have all that extra stuff.
Okay, no, it was more like, can these induce apoptosis as well, or no?
These?
Yeah.
No.
Okay.
So I have a potentially dumb question. So you mentioned, so like with these, like the 1 through 6, you did the fast and the slow, and you changed up, like you alternated them to change if they were fast and slow. Is it possible to add like the anchor that 7 has onto the others?
Great question. And people have done that, and so they've added to P2X2, and 1, and 3, I think. And it changes it, but it doesn't keep it completely open, okay? It does not have the same property. So the way I interpret that is, it's necessary to have the cis anchor there, but it's not sufficient. It needs to be palmitolated. So, of course, nobody proved this because people did this before I solved the structure, and they made all sorts of, nobody was talking about palmitolation. But what I think has to happen is, you can't just have the residues for the anchor, it has to be palmitolated. So whatever palmitoyltransferase recognizes P2X7 receptor to palmitolate those residues probably can't recognize P2X. We don't know what the recognition sequence is, but if you just take those 18 residues and stick it in P2X3, it's probably not going to get properly palmitolated. That's how I interpret that. But people have tried that, I think 10 or 12 years ago. Gennard, I think, was the name of the first author on that paper. And I looked at that, and they made some interpretations and had nothing to do with palmitolation, and they kind of glossed over it. But they also, people noted that these 18 residues are only in P2X7, so they tried to put it in all the other residues to see what, all the other subtypes to see what it might do. And it didn't seem to do all that much, but I think that's because it's not getting palmitolated. It needs to be palmitolated.
I mean, but like the rest of the cytoplasmic region, or like, residues don't need to be palmitolated for their necessarily apoptosis-inducing function, right?
The channel has to stay open.
Oh, okay. But like, so are any of these more like sort of faster proteins, have they added that cytoplasmic domain to them? Or is that more?
Oh, that's an interesting question. You mean like create a Franken receptor where we take the whole cytoplasmic domain and add? No one's done that. Okay. Yeah, that's a good that's something we could try. I don't know if it will fully fold, but yeah, it's a great thought. Yeah, no one's done it. Yep.
I want to go back to the forest and trees because is P2X a generic design that a platelet agglutinating thing is going to use a P2X, a specific one, or I mean you looked at the applications early on.
Yeah.
So it does this. Would they be different
subtypes? Yes.
So we're talking about different tree structures here for a very specific study that you're playing with. I guess my question is, what are these relating to?
Oh, so P2. Okay, so I'll tell you.
Is that cardiac?
So I'll tell you. Let's just talk about P2X7 receptor because that's the one we're most farthest along with. I was contacted by these collaborators, one in particular in Europe, who is a synthetic chemist. He wanted to develop novel antagonists against P2X7 receptor, and we're solving the structure of them bound, and we're trying to improve the quality of the ligand, and they are interested in neuroinflammation, okay? The neuroinflammation model. And what they're seeing with some of these compounds that we've developed is they have a mouse model of neurodegeneration. And the compound that we've sort of developed, he can feed, it's bioavailable, he can put this compound in the feed of these mice, or in the water of these mice, and they're actually seeing the improvement in the memory and a slowing of both short-term and long-term in these animals. And so that's one application.
Right, but that's not interfering with pain reception, that's not interfering with hearing or anything else. So these are very specific P2X
that's for
molecules.
P2X7.
Yeah, right, okay. I guess with that, how do the different P2Xs or P2X, whatever, vary from each other? So in other words, I guess
So the goal is to design
target something for a very specific P2X7.
Correct.
receptor.
The goal is to define to design a molecule that binds to P2X7 1,000, 10,000, 100,000 times more selective than P2X1 or P2X2 or P2X3. So it doesn't inhibit those receptors.
Right, but a P2X7 receptor in the brain and whatever neuroglial cells or something else, as opposed to cardiovascular or other peripheral nervous system receptors. That's why I'm asking how unique each of these different receptors would be in different body parts potentially.
Oh well, I mean, so I think I understand your question. P2X7 is P2X7 is P2X7. So it's going to depend on if you can. So for example, it tends to be that they're on circulating macrophages. P2X7 is on circulating macrophages. So and monocytes. So yeah, you're right. You're exposing all of them to your ligand. That's how any molecule works, right? Unless you get targeted therapy.
Right. My arm will have no neurological degeneration or dementia.
Sure. Yeah. Yeah. Well, I'll tell you. One of the things they're also doing in Europe is trying to in animals inject directly into the brain. Yeah. But obviously that's not ideal, right? In any condition there are risks, and like as a doctor when I prescribe any drug there are risks and there are benefits, right? And the idea is your benefit outweighs your risk and you try to minimize your risk, right? Yeah. Yeah. Yeah.
I have a slightly extraneous and very undergraduate question.
Very well.
Undergraduate.
Oh, sure.
How do you go about starting research?
In a pandemic.
In a pandemic, yeah. You have mentorship, right, and help. And so, I just so happened that Dave Farron's my PhD mentor. I started in his lab when he was just starting out. So I saw what he went through, and I remembered it. I still have some notes. My goal was to be able to start my own lab someday, and so I have notes from that experience and I have memories from that experience. And then you do a postdoc, and then you get information from that experience as well.
Yeah.
That's what I would say. But it's not easy. Anybody else? Yeah.
I think I saw a graph where P2X5 seemed to remain in the apo state.
Yeah, no, P2X5 is a really interesting one. So P2X5, there are two forms of P2X5. There's a so-called ancestral form, which seems to be present in African American and Middle Eastern populations. And then there's the modern form, which is more present in kind of Caucasians and everybody else. And P2X5, sorry?
Nothing.
No, P2X5,
We had this discussion earlier.
yeah. P2X5 is missing about 10 or 12 residues, I can't remember the exact number, in transmembrane domain 2. So they think, the hypothesis is it's not functional in many people, but it is in some people. And so that's kind of interesting, right? It depends on what isoform you have. And who knows, it may explain why, things like this may explain why certain populations of people are more prone to certain diseases, right? So yeah, but P2X5 is a weird one. P2X6 is another really weird one. It allegedly or supposedly doesn't form P2X6 homotrimers. It only forms random heterotrimers, not random, several isoforms of heterotrimers. So nobody understands anything about it because we don't understand anything about heterotrimers. Right? Anybody else? Good. Yeah, well, yeah. One more. Maybe one more.
Okay. I was just wondering, are are the different types of receptors ever co-expressed on the same cell, or
Oh.
is it always one cell, one type of receptor?
Oh, great question. That's a good question. No, I would say there's co-expression for sure. Yeah. I think there are certain ones that are more dominant, like in monocytes and macrophages, for sure P2X7. Platelets, only P2X1 really. Vascular smooth muscles, P2X4, but it's got some 1. But they are co-expressed, but there are dominant forms in certain tissue types.
So targeting one receptor won't completely knock out function in a given cell then?
Probably not completely. Probably not completely. But they may do, it's unclear. They may do, different subtypes might do slightly different things in any given tissue, right? We don't know.
Where do you find P2X5 and 6?
Top of my head, 5 and 6 I've studied less. I don't know. Adam, do you know?
I don't know.
I'm sorry, Aaron. We've just started perusing around 5. 6, we attempted some work on it, and it's going to be hard to do because it just doesn't form it. We can't. We have no way of studying heterotremors yet.
There's one really good review article with just a list.
Yeah, it's all over.
I mean, has anyone done that sort of like historical genetic analyses on like the development of P2X6 and 5 then? Or?
There are a couple of interesting papers. What's that? Yeah. So there's this like, what is that? Some kind of crustacean? No, zebrafish or something. There's some kind of like oceanic organism that has. So what was really interesting about this part of the molecule, this ballast, is when I solved this structure, I had to build that from scratch because there are no there were no, at least in mammalian and eukaryotic cells, there were no structures that had homologous shape to it. Zero. There was there's one sequence in some zebrafish, some nanoprotein, that was loosely similar, but no structure, so I had to build it from scratch. And so at some point maybe P2X7 picked this up from this oceanic organism. But I don't know nobody's really talking about it. I don't know much about the evolution of P2X7 receptor. But it is very interesting that there was just no sequence homology and zero structural homology to anything in the database. So we named the ballast fold. So, yeah.
That's super cool.
Yeah.
I know you just said you don't know much about heterodimers.
Yeah.
But if,
Trimers.
heterotrimers. But if a P2X7 molecule were to heterotrime or to bond with other ones, would that be enough polymerization to cause?
That's a great question. That's a great question. I don't know, but it does. It supposedly does form a heterotrimer with P2X4, I think, right? And then, but here's another really weird thing. There's an alternatively spliced version of P2X7 receptor that is missing the cytoplasmic domain, and it is expressed in some cancers, overexpressed in some cancers, right? And so you can't initiate apoptosis, right? So yeah, it's very, very interesting. So it's cardiovascular system, neuroinflammation, and then a number of cancers. All right.
I think we got
Thank you all for putting up with the chaos.
What chaos?